Today, we are going to talk about cell division in germline cells, also called meiosis. As we know that all of the cells need to divide for growth, for regeneration, and for reproduction. There are two types of cell divisions: mitosis, in which the cell, one cell, also called a mother cell, divides into two daughter cells, and these two daughter cells have all the properties just same as that of the mother cell. The other type, and this type of division, mitosis, occur in the somatic cells, all cells of the body other than the germline cells, the gametes. But in the germline cells, the gametes, for the production of gametes, the other type of cell division occur, which is called meiosis. Meiosis is a cell division which occur in the germline cells to produce the gametes for sexual reproduction. Meiosis is also called reduction division, because in this type of division, the chromosome number of the mother cell, when it divides into cells, becomes 1/2, exactly 1/2. Meiosis occur in the germline cells to make gametes, sperms, and the eggs. Meiosis is a reduction division in which the chromosome number becomes 1/2 and one cell divides into 4 cells ultimately. And the end of meiosis, unlike mitosis, the one cell divides into 4 cells. And all of these 4 cells have a half chromosome number in comparison of the mother cell. For example, human beings have 46 chromosomes in total. These 46 chromosomes are present in 23 pairs. In each pair, the 2 chromosomes which are present are just alike in their generalized appearance, though there are certain differences in the set of genes, because one part of gene on one of the member of the pair may be a dominant one, and the gene present on the other one may be the recessive one, or both are dominant, or vice versa. These pairs are called homologous pairs. Homologous because they look alike in their general appearance and shape, particularly when the cell is dividing, because we already know that the chromosomes at interface are very thin and they are not visible. These chromosomes are visible only when the cell is dividing, maybe during mitosis or maybe during meiosis. So when there is a division phase, the chromosomes appear in the pairs. We call these pairs the homologous pairs, and we call these individual chromosomes, that is 2 in a pair, homologs. We call this chromosome number, the total chromosome number, for example, like 46 total chromosomes in human beings, 2n. When the cell divides by meiosis into 4 daughter cells, this 2n number of chromosomes becomes 1/2. That is, now each daughter cell have exactly half number of chromosomes as the mother cells had. We call this number n. And the parent cells, the mother cell, which have that 2n number, we call that cell a diploid cell. Diploid means that it have 2 sets of chromosomes. And the daughter cells, the sperms or eggs, they are called haploid. They have a n, that is a half set of chromosomes. So we call them haploid. The mother cell is called diploid. The daughter cells are called haploid. Which means that they have a exact half number of chromosomes. Sometimes we call this ploidy. Animal cells and many of the plant cells, they are diploid. Their germ cells are diploid, and when they divide for a sexual reproduction into sperms and eggs, they become half in their number, divided by meiosis, and we call them haploid. But in some plant cells have even more sets of chromosomes. For example, if they have 18 chromosomes, these are divided into 3 sets. We can call them triploid. That is, triploidy, 3 sets of chromosomes. There are different types of plants which even have even more sets of chromosomes. We call them polyploids, and we call this phenomena polyploidy. Now, to understand this phenomena, just look at a diagram. You can see a set of chromosomes. These two are actually the homologous chromosomes. When they are not duplicated or replicated during the S phase. As you can see, after replication or duplication, the contents of both of these chromosomes will be duplicated, and now each one chromosome consists of 2 sister chromatids, and the other part, the other homologue, the other chromosome, which is part of the pair of this homologous chromosome, consists of now 2 other sister chromatids. This is the reason that we call them diploid, have double number of chromosomes. Now then what happened that when the gametes are formed, the sperms and the egg, they have to join together, each from one parent, and when these cells join together, which is called fertilization, after this process, the sperm have a half number of chromosome, n number, and the egg have a half number of chromosome, n number. When they combine, the normal 2n number of chromosomes, or we can say that diploidy is retained in the zygote formed, and the next individual, the embryo which is formed by fertilization, again have the same number of chromosomes 2n as its parents did have. But this process of meiosis actually offers a great number of variations, because half set of chromosomes are coming from one parent and the other half is coming from the other parent. The result is lot many changes, or lot many different sets of genes which may result in different characteristics. Now look at another diagram. This diagram shows the set of chromosomes in the human beings. If you look at the diagrams, it shows the chromosomes in pairs. You can see in the pair 1, there are 2 chromosomes. Just like that, in the pair 2, there are 2 other chromosomes which have different forms. But in the pair 1, both chromosomes, they look like each other. Just like that, in the pair 2, they also look like each other. Just like that, the chromosomes 3, 4, 5, 6, and so on, up to 22, they are alike. Just the one pair, as you can see the last pair in the right corner down, you can see that there is an X and there is an Y. This only set, the XY pair, is different in human beings, in the males and females. Males have a set of XY. So in this pair, the chromosomes are not homologous. They are not alike. Y is different from X. But in females, this is again a homologous pair, which makes X. So in females, all the pairs are homologous, but in males, one pair is not homologous. Now, we talk about the meiosis. Meiosis occur in the germline cells, in the ovary of the female or in the testis of the male. There are the germline cells divide into 4 daughter cells, each, as we know now, haploid in number. That is, have a half number of chromosomes. Meiosis is also called a reduction division because chromosome numbers become 1/2 in each daughter cell. Meiosis consists of 2 stages, Meiosis I and Meiosis II. In Meiosis I, actually, Meiosis I is technically the reduction division, because during Meiosis I, the chromosome number becomes 1/2 and 2 daughter cells are formed from 1 mother cell. The Meiosis II occur in both of these daughter cells separately, and both of these daughter cells, each one divides into 2 further. So ultimately, at the end of meiosis, we have 4 daughter cells. But the division, the actual reduction division in which chromosome number reduced to 1/2 is the Meiosis I. Meiosis II is just like that of mitosis. Now we talk about Meiosis I. Meiosis I consists of a prophase. We call it Prophase I because this is the prophase of the Meiosis I, Metaphase I, Anaphase I, Telophase I, and a cytokinesis. When the cell divides into 2, then when the nuclear division is complete, then the cell divides into 2 by cytokinesis, and when it is almost divided, it starts its next phase of division called meiosis II. Meiosis II is just like the mitosis. It simply divides both the cells into into same type of cells with equal number of chromosomes. Now we look at the detailed process of meiosis. Meiosis I. Meiosis I consists of prophase, then metaphase, then anaphase, then a telophase. We look at first of all the prophase I. As we know that the chromosomes, they are just like mitosis, they are already duplicated. It means that every chromosome have two sister chromatids. It is doubled in the number. As you can see in the diagram, there are two homologous chromosomes which are shown. Each chromosome, which is now called each homologue will consist of actually two sister chromatids. You can see one in green and the other in blue. The green one is one homologue, that is one chromosome which is duplicated. The blue one is its homologous partner, and which is in its appearance just like that of the first one, which is shown in blue color, and this also consists of two sister chromatids. Now what happened in the prophase I, as is shown in the second diagram? The prophase I is the longest phase of meiosis. In this phase, actually, the homologous chromosomes, they pair up with each other. They come close together, and some of their parts attach to each other to make the structures called chiasmata. In the diagram, the second diagram, you can see a single chiasma in which one part of homologue 1 is attached with the, or we can say crossed with the part of the homologue 2. We call this process pairing up or synapsis. We say that these chromosomes, the homologues, they are synapsed together. But remember at this stage that only the homologous chromosomes will pair up with each other. As we know that in human beings, for example, they have 46 chromosomes, and there will be 23 pairs. The chromosomes in one pair only will make chiasmata with each other. One homologous chromosome will not pair up with any other homologous set. They will only pair up with their own type. If they do so wrongly, that will be an error in the meiosis, which may result in a problem later on. So usually or normally, homologous chromosomes pair up with each other. We call them these pairing synapses. Now what is the purpose of this pairing? Purpose of this pairing is recombination. These parts of chromosomes which pair up, which are crossing each other, what will happen that these parts, both of these parts, will break up from their original chromosome and will attach to the other homologue. As you can see in the bottom of the diagram, now the two homologues are shown, but you can see that the homologue on the top in the green color have a small part of blue color, which is actually broken up during the chiasmata, during the synapses from the homologue 2 and attached to the homologue 1. Same happened with the homologue 2. You can see in the last part of the diagram that the one part of homologue 2 looks like green, because actually this is the part of homologue 1 which was during chiasmata formation, again the pairing up, broken up and attaches to the homologue 2. Now you see that both of the chromosomes, they have a large part of each other. They exchanged their parts. We say that they exchanged their DNA or nuclear material. The result of this situation is new recombinations. There are new sets of genes which are formed, and the result will be new characteristics which will appear in the organism. This is the most important property and advantage of the sexual reproduction, that during the meiosis process, the cells, the chromosomes, they do recombine. And as we know, as many chromosomes as an organism have, as many recombinations it can form. And every recombination may result into a different characteristic, which gives an evolutionary advantage to that particular organism. Meiosis, the prophase I is one of the most important parts of meiosis. These paired chromosomes, which are making pair, as you can see in the diagram on the top, these chromosomes, homologues which are paired, the homologue 1 and homologue 2, collectively are called sometimes bivalents or tetrads. They are called bivalents because both homologues are attached with each other. They are called tetrads because we know that due to duplication or the replication, each homologue consists of now 2 sister chromatids. So in total, they are making 4 chromosomes. We call them tetrads, or sometimes we call them bivalents. And till end of the prophase I, lot many recombinations like this are formed, constituted, because almost every homologous pair up with each other, and they exchange their different parts. So lot many recombinations they are formed. With this, all the other features of the prophase, they also takes place. The spindle fibers are formed because chromosomes have to attach to spindle fibers to move from their place, from the center towards the poles. So spindle fibers are formed. In animal cells, centrioles will do it. In plant cells, spindle fibers will be formed by other methods. And the nuclear membrane, ultimately, at almost end of this phase, will disappear. Now there is one more important difference. Now each homologous pair is attached with each other by one kinetochore. There is one kinetochore for each homologous pair, which is in contrast to mitosis, in which there is one kinetochore for one sister chromatid. Now both sister chromatids of one homologue are attached with only one kinetochore. And this is very important because we know that meiosis is the reduction division, and one homologue have to go to one pole and the other homologue have to go to the other pole. Now we know that at about end of the prophase, spindle fibers which are already formed, each homologue attaches to the spindle fibers with its kinetochore. Now, because there is one kinetochore per homologue, so every homologue as a whole is attached to a spindle fiber. Which is same is true for the other homologue. There is one more important thing. As we know that every organism which is sexually reproduced always have one set of chromosomes coming from the mother and the other set coming from the father. The homologues which are coming from mother and father, they are arranged in different orientations. That is, in one pair, for example, the maternal homologue will be on one side towards one pole, and the paternal homologue will be on the other side. This is different for each chromosome. It means that in a gamete, there are about 50-50 chances that the gamete will have a maternal homologue or a paternal homologue, which is also a very important feature for, we can say, new recombinations. And at the end of when the spindle fibers are formed, the chromosomes they attach, the nuclear membrane is dissolved or start disappearing, and it is disappeared at the end of very, very long phase, the prophase I, and then the prophase I ends. When the prophase I ends, the next phase starts, that is metaphase I, because this is part of the meiosis I. Now in the metaphase I, the chromosomes, the homologues, they are attached on the spindle fibers, arrange themselves on the equator. All of the homologues are arranged on the equators almost in a rough straight line or in rough circular, we can say, part. After the metaphase comes metaphase I, comes the anaphase I. Now anaphase I is very, very important because in this anaphase I of the meiosis, the chromosomes have to move towards the pole, and now half chromosomes have to go to one pole and the half chromosomes have to go to the other pole. What happened that because we know that there was one kinetochore per homologue, per two sister chromatids. It means that the two sister chromatids are going to move towards one pole and the other two sister chromatids are going towards the second pole. Now the microtubules or the spindle fibers, they start pulling the chromosomes towards the poles of the cell, now because again I repeat the same thing, because there is one kinetochore per two sister chromatids of one homologue. So one homologue, which consists of now two sister chromatids, will move towards one pole, and the other homologue, we can call here homologue 2, which also consists of two sister chromatids, will move towards the other pole. So the half set of the chromosomes is moving towards one pole and the other half set is moving towards the second pole. So this is the anaphase I. Now this stage is extremely important because if there is a slight error in this phase, it results into an unequal distribution of the chromosomes in the gametes. And if there is an unequal distribution of chromosomes in the gamete, it results in very, very serious disorders. When the anaphase finishes, the chromosomes are moving towards poles, almost they are reaching near poles. The last stage of meiosis I starts, that is called the telophase I. In telophase I, the half number of chromosomes reached at one pole, the other half of chromosomes half set is reached at the second pole, and now the nuclear membrane starts forming and the chromatin material start slightly decondensing. With this, the meiosis I, or the nuclear division 1 of meiosis, is complete, and then the cell is divided into two cells, in again just like in plant cells by a phragmoplast, and in animal cells by a cleavage furrow formation. Cell is divided into two. Now there is a slight difference from mitosis. That what happened that sometimes these cells are completely divided into two. When they are just dividing at the end of their cytokinesis, they just start their second nuclear division, which is called Meiosis II. Because meiosis is a continuous process. So what happened that when the nuclear division is complete, cleavage furrow is formed, and cleavage furrow almost reaches at the end, then the next nuclear division just starts. We call it Meiosis II. Now we have a look on a diagram which explains the Meiosis I. If we look at the diagram, the first part shows the interphase I. When the cell is at interphase, its chromosomes or chromatin material is not very visible, and it is showing a centrosome, a pair of centrioles. The next diagram towards right, you can see that the chromosomes, the homologous pairs, they consist of two sister chromatids each, and they are forming chiasmata. Now here in this diagram, you can observe that per homologue, more than one chiasmata could be formed. You can see in the small sized chromosome pair, the homologue pair, there is one chiasmata. But you can see the chromosome which is larger one, there are two chiasmata. So it means there may be more than one chiasmata per homologous pair. Then comes the next stage, the metaphase I. In the metaphase I, as we talked about it, in it the chromosomes are arranged on the equatorial plate in the center, but now each homologue is attached with the kinetochore to the microtubules, the spindle fibers, and not sister chromatids. In anaphase I, you can see that the whole homologue, the complete chromosomes with its both sister chromatids, is moving towards the poles. And you can observe various recombinations present in these chromosomes which are going towards the poles. Then comes the telophase, they start decondensing, nuclear membrane starts appearing, and the cell divides. But when the cell just divides, the meiosis II starts. As you can see in the diagram, because now we know that after meiosis I, one cell is divided into two cells, each with a half set of chromosomes, each with a half number of chromosomes. In case of human beings, for example, if there were 46 chromosomes in 23 pairs, then each daughter cell after meiosis I now will have 23 chromosomes, one from each homolog. In case of an organism, for example, which have a 16 total chromosomes in 8 pairs, both daughter cells will now have 8 chromosomes each. That is one set, and all are recombined. So recombination is done. Now both of these cells will enter into the meiosis II. Meiosis II, so it means that meiosis II will occur in the cells, in the two cells which are already produced by the meiosis I. You can see the first cell is dividing and the second cell is also dividing. The meiosis II is just like the mitosis. In the meiosis II, first phase is prophase. We call it Prophase II because this is the prophase of the meiosis II. In Prophase II, the chromosomes, they appear, they become visible under the microscope because of the condensation, because they are condensed, they are thickened, and the centrioles start moving in the animal cells. Centrioles start moving towards the poles and they start making the spindle fibers. And again, at the end of the prophase, the nuclear membrane will disappear. At about end of the Prophase II, the nuclear membrane disappears and the chromosomes and spindle fibers are there in place in the center. After the end of Prophase II, metaphase II starts. In the metaphase II, the chromosomes now arrange themselves on the equatorial plate. Now, as we know, they are duplicated, and now each sister chromatid is attached with the spindle fibers with the help of separate kinetochores, like that of mitosis. And they arrange themselves on the equatorial plate. As you can see, this is observable in both cells, that they are arranged on the equatorial plate, and now each sister chromatid is separately attached with the spindle fiber. Then comes the anaphase II. In anaphase II, the chromosomes start separating from each other because the spindle fibers, they start pulling these chromosomes towards the poles. And now, because each sister chromatid was separately attached to a spindle fiber by its own kinetochore, each sister chromatid moved towards the poles. So the result is an equal number of chromosomes distributed towards each pole. When the anaphase II ends, then comes the telophase II. In the telophase II, the chromosomes in equal numbers now reached at the poles. Nuclear membrane starts forming. Nuclear material start decondensing again and become invisible as that of the interphase. The spindle fibers, they are deformed, they are degraded, and each cell is divided into two, makes total of the 4. So in meiosis I, what happened? That one cell is divided into two, each with a half number of chromosomes, but both of these cells, which were the products of the meiosis I, were individually divided by another phase of meiosis called meiosis II, and the end products of these two cells is 4 cells, all with a half number of chromosomes, because reduction is already complete in the meiosis I. So this is how, by one mother cell in the germline cells is divided into 4 cells or daughter cells, either sperms or eggs, which have a half number of chromosomes in each. There is another difference in the meiosis between the males and the females. In males, one germline cell divides into 4 equal sized cells. It means that one germline cell makes 4 sperms, all with a haploid number of chromosomes, n number of chromosomes. But in females, there is a difference. As you can see in the diagram, when after the meiosis I, which is the reduction division, chromosome number become 1/2, But when the cell divides, it divides unequally. It means that chromosome number is half in both of the cells, but the cytoplasm and the cytoplasmic contents goes more towards one cell, and the other cell, though have equal number of chromosomes, but have fewer parts of cytoplasm and the others, which results in one cell which is large, which is called a secondary oocyte, and the other is called a polar body, which is very small. Next division, in the next division, the polar body again divides, but it divides, that is in the Meiosis II, it divides into equal size cells. But the secondary oocyte, that is the other cell, which is larger in size, is again divided by Meiosis II, but this division is again an unequal division. The result is in a very large ovum and a second polar body. So it means that in a female, the cells, as you can see in the diagram, one cell, the primary oocyte, is divided into 4 cells, but 3 of these are called polar bodies, which are small in size and which are discarded, and the one which is larger in size is called the ovum, and this is retained for fertilization. So there is a difference between the meiosis in males and females in this regard. Now we do compare the processes of mitosis and meiosis. As we know that mitosis divides the cell, one cell, somatic cell, into 2 daughter cells. But in meiosis, one cell, the parent cell or the mother cell, is divided into 4 daughter cells. In mitosis, this is alike in males and females. The somatic cells of males and females, or every type of cells, their division in males and females is not different from each other. They are just alike. But meiosis is different in males and females because 3 are polar bodies and 1 is an ovum in the females, but in males all of the 4 are sperms. Then in mitosis, the chromosome number remains same. In the daughter cells, we call it 2n. After mitosis, 2n number of chromosomes is retained. But in meiosis, the chromosome number is reduced to 1/2 in the daughter cells. Either these are 4 or 1. The chromosome number is 1/2 in the gametes. So mitosis and meiosis, they are different from each other, and mitosis occur in the somatic cells, meiosis occur in the germline cells. Now we talk about the importance of meiosis. What is the importance of meiosis? The most important feature of meiosis is the genetic variation by recombination. As we know that the natural environments in which the organisms are living, they are always changing. The temperatures change, sometimes earthquake comes and the whole environment is changed. There are different types of environmental factors which always they are modifying. The availability of foods, the availability of shelters, and everything changes, and the organisms have to modify according to these environments. These recombinations by the crossing over in the prophase I of the meiosis provides the basis of new recombinations. And this provision of new recombination makes new characteristics. These characteristics, if are suitable for the organism and makes it more fit for the environment, the organism's survival is improved. So usually the organisms have a better survival if they have new good characteristic. But it could be otherwise. If the characteristic which appears is not good, then the survival will be reduced. And as we know that the Darwin's theory says that the fittest will survive in an environment. The second benefit is this, that because of meiosis, half number of chromosomes go to one gamete, and then the gametes have to combine. And during as we know from the anaphase, because we covered anaphase, in anaphase, this is not necessary that whole set coming from the grandparent will go to just gamete, just one gamete. There will be a mixture, because in anaphase the chromosomes are arranged in homologous pairs, and in each pair this is not necessary that the paternal chromosome from father will be on one side and the chromosome from mother is on the other side, but this may change. So there is a 50-50 chance that the gamete will have the paternal chromosomes or the maternal chromosomes. So in each gamete, there is a mixing or combination of few paternal and few maternal chromosomes. This provides another degree of recombination. This is the most important feature of meiosis. This is the importance of meiosis, the genetic variation, which makes usually the organisms more fit to survive in the changing environments. Just have a look in the diagram, which shows homologous pairs and then shows a recombination, a chiasmata formation at two places. And this chiasmata formation at two places, as you can see, if we look at their genes, in the initial chromosomes, there was a combination of capital Y, small y. For example, in a plant, this is for yellow and green. These are the genes for yellow and green seeds. After recombination, we can see that the first one will become yellow, the second one may become a green, the other one may become due to a recombination and in between color. So a genetic variation arises due to these recombinations in meiosis. Now what happens if an error occur in meiosis? Errors in meiosis could have serious consequences or disadvantages. Usually, errors in meiosis comes from non-disjunction. To understand the non-disjunction, we have to look at a diagram. As we know that meiosis have two phases, meiosis I and the meiosis II, which results in formation of gametes. As you can see in the first diagram on the left, in the anaphase II, when the chromosomes have to move from the center towards poles, if the homologous chromosomes are not separated in anaphase I, then what will be the result? That the whole homologous pair will go towards one cell and the other cell will not have any chromosome on its place. So that will be empty for that chromosomal pair, that whole homologous pair. Then these two cells will divide by meiosis I, they will result in unequal formation of gametes. As you can see, in the first one, it shows an n+1. That is, one extra chromosome is here. The second also have an n+1. But the other one, where no part of homolog went, the chromosome number is n-1. That is, one chromosome less than the haploid number. And the fourth one is also n-1. That is, one chromosome less than the normal haploid number n. This is called non-disjunction. That is, in anaphase, the homologous set which should naturally or normally separate are not separated, but they moved towards one pole only, and the other pole did not receive any chromosome. This non-disjunction could occur in the meiosis II as well. Sometimes what happens that meiosis I completes correctly, and both of the cells, which are the result of meiosis II, they have the correct number of chromosomes. And then they start with the meiosis II. But at anaphase II, when they have to separate, as the diagram on the right shows, when they have to go to the poles, in one of the cells, division is normal and the chromosome number is n. But in the other cells, which is divided into two, one chromosome with both of its sister chromatids move towards one pole. And on the other pole, there is no chromosome. Because one sister chromatids, as normally, as you can see in the last right diagram, should go towards one pole, and the other sister chromatid should go towards the other pole. But what happened? That both sister chromatids keep attached to each other, did not separate, and they move towards one pole only. And the other pole remains blank. The result is, again, we call it a non-disjunction, no separation, and the result is one gamete, which is n+1, that is haploid number plus one extra chromosome, and the other one is n-1, that is haploid number with one chromosome less. Now, as you can observe in these two diagrams, and if you compare both of these non-disjunctions, the first one, non-disjunction in meiosis I, and the second one, the non-disjunction in meiosis II. If we compare those, we can see that the non-disjunction in meiosis I is more dangerous, because the result is all the four gametes have abnormal number of chromosomes. But in the case of non-disjunction in meiosis II, we have two normal gametes and two abnormal gametes. So it means that if non-disjunction occur in the meiosis I, then the result will be all abnormal gametes, more dangerous. But in case of non-disjunction at meiosis II, we will have two normal gametes and 2 abnormal gametes, that is 50% correct and 50% incorrect. So this is non-disjunction. And what will be the result? If these unequally distributed chromosomes in the gametes will combine with a normal egg or sperm and will be fertilized to make a zygote, the individual which is formed will not be normal. This results in different types of diseases at birth. You can see here an individual in the picture with a Down syndrome, which is a mental disorder. The child have inabilities, intelligence inabilities, and have many other problems. And the reason is that if you can see in the yellow circle, the chromosome number 21 have, rather than 2 normal parts of a homolog, it have 3 homologs. Just non-disjunction in 1 chromosome only out of 23 pairs. In just 1 pair, there is a non-disjunction. And the fusion of the gametes, one gamete from normal parent, the normal gamete, and one gamete from a parent where a non-disjunction occurred, which have an extra chromosome. The result is a very serious disorder, Down syndrome. All the chromosomes you can see, the 1, 2, 3, 4, 5, 6, 7, 8 are normal. The last is also normal. Just at 1 chromosome, there is, we call that 3 chromosomes are present in the set. Rather than 2 homologs, there are 3 homologs which are present. It means that a slight disorder, a slight problem, a slight non-disjunction, just even in 1 chromosome, may result in a very, very serious disorder. And there are a lot many other disorders which may occur due to non-disjunction at different types of chromosomes. So errors in the meiosis may result in serious inborn disorders. And as you can see here, it's a very serious mental disorder, and person is unable to run the normal life in normal circumstances, and they need special attention. Now the last topic for today, the twins. Twins, we know, if we look in our lives normally, many pairs of the twins, 1, sometimes 2 pair, maybe 3 or more. There are 2 types of twins. One is called fraternal twins. The other are called identical twins. We see twins are in our normal life. Many times we see that they are just like each other in their height, in their face, in their color, in their features. Everything is just alike, and they are of same gender. Both are male or both are female, both boys and both girls. But sometimes we see the twins which are actually very different from each other and who don't look like twins. They have different heights, they have different features, they have different intelligence, different other characteristics. Actually, according to the divisions, there are 2 types of twins. One is called fraternal twins. Fraternal twins are those twins which look to us different from each other, just as the siblings are different from each other, which are born at different times. What happened that they are actually produced by different eggs. Sometimes more than 1 eggs are produced by the ovary, and both eggs are separately fertilized. The result is 2 genetically different individuals. They are as different as those individuals are which born in different times. But the identical twins, they are produced by the division in the same egg. What happened that an egg is produced with the haploid number, but it somehow divides by mitosis into 2. And when the egg is fertilized, after its fertilization, it divides into 2, which means that zygote is divided into 2. We also call them monozygotic twins. Because they are product of same zygote, they have exactly same genetic features. They have exactly same type of chromosomes. So they look just as each other. They must have same gender, both are boys or both are girls. They are identical thoroughly in their genetic recombinations and in their genetic characteristics. But there is another thing which may makes them different. As we know that characteristics of an individual, particularly its behavior, does not only depend upon the genes. Genes play their role, but the interaction of environment with the genes or we can say the gene-environment interactions, make certain characteristics, particularly the behavioral characteristics and the intelligence. By various studies in different parts of the world, shown that if identical and monozygotic twins, they are grown up in different situations and conditions, they develop different types of intellectual skills, they develop different types of behaviors according to the environment in which they are grown up, they are brought up. It means that genes alone are not responsible for all the characteristics of an individual, but the environment plays its role, and the environment and genes, both of these, interact with each other to produce a full individual, a complete individual with all its characteristics. Life is a production of genes in interaction with the environment. So this was all about meiosis, its significance, its errors, the results of its errors, the twins, interaction of the genes and environment. I hope you understand what we talked about today. We will see each other in the next lecture. Thank you.